Suppose you have sequenced the genome of two animals from different species, a lion and a cat. Now you want to find out how similar these two species are to each other. How would you go about this? The first step you would probably perform is called a sequence alignment. If you have two sequences, an alignment of these sequences tells you which parts of one sequence correspond to which parts of the other sequence. For example, with these two sequences, a good alignment would match up the subsequence CAT within the lion sequence and the sequence CAT. For example, we could do it like this and just write these two sequences on top of each other to visualize where the overlap is. A position within an alignment is also called a site. Every character corresponds to one site. At each site for each sequence, there can either be a character, for example, this A at the beginning, or there can be a gap. The second sequence in this alignment has 5 gaps to make up for the difference in the number of characters between the two sequences that we are aligning. This here is already our first sequence alignment. It isn't always as easy as in this example, and that's why many different methods and algorithms have been developed to do this correctly and efficiently. In this video, we will only be talking about pairwise alignments. Pairwise alignments are alignments between exactly two sequences. There's also something called a multiple sequence alignment, or MSA for short, which consists of more than two sequences. This is really important for a lot of modern deep learning methods, for example, in protein modeling. And we'll get to this in another video. But for now, let's start with pairwise alignment. Within pairwise alignments, there are again two different types, namely the global and the local alignments. A global alignment aligns two sequences from beginning to end, while a local alignment only aligns subsequences, small snippets within the sequences. In this video, I will discuss a simple method that helps you visualize possible alignments, called the dot matrix method. Let's say you're just getting started with your analysis of the cat and the lion genome, and you have two short sequences, maybe of a particular protein. Now you want to get an intuitive understanding and visualize the similarity of these two sequences. Knowing how to use this method would also help us in understanding the more complicated but widely used other methods. Last and Needlemann-Wunsch. As the name suggests, you start with a matrix or a table that contains the two sequences you are comparing. You write one of the sequences horizontally and the other one vertically. Then you fill in the matrix by placing a dot in each square for which the character in the column and in the row match up. For example, looking at the first square here, we can see that the letter in the column and the row do not match up, so we don't place a dot. Same holds for the second square. However, the third square has a C as the column character and a C as the row character. Therefore, we place a dot in this square. The same goes for the fourth square and also for the second to last square. In the squares without matches, you can either put a cross like I did here, or you can also simply leave them blank. Let's look at the second row. Here, the row and column match up in the first square and in the fifth square. The other ones do not match up, as there's no other A's in the lion sequence. Finally, we complete the third row of our table. Once we've filled in all of the squares, we can look for patterns in this matrix to identify possible overlaps or similarities between the sequences. To make this a bit easier, I will clear up the crosses. Okay, very good. We already see an important pattern here, a diagonal. Not surprisingly, this corresponds exactly to the subsequence that matches between the two sequences, the CAT or cat sequence. This helps us in finding an alignment by hand and without needing to do any programming. However, it won't always give us a good result where we can immediately infer the correct alignment from this matrix. But even if it doesn't, it can still give us some information about how the sequences are related. There are 3 different patterns to look out for. The first one are indels, or insertions or deletions in the sequences. So let's say we have a sequence A, T, C, C, A, T. We compare it to the same sequence, but we include an insertion of G, G, G. Now, let's fill in the matrix. As before, we put a dot whenever the column and the row character match up. Nice. So we've now filled in this whole matrix, but we can see that there is a big gap in the center of this matrix. These three columns do not have any dots at all. And these are also the columns with the inserted characters. Therefore, the dot matrix method can help us see insertions or deletions quickly. In this specific example, the character G doesn't show up anywhere in the vertical sequence. Therefore, there are no dots at all. However, even if the character showed up somewhere in the sequence, there would at least be a gap in this diagonal pattern. The second pattern we can easily identify with the dot matrix method are repeats in the sequence. I've modified the vertical sequence to include the word cat twice. Now let's see how that affects the matrix. We again include dots in every square in which the column and the row match up. There are several repeating patterns within the matrix. So this 3x3 grid here is repeated directly below with the same structure of dots, and the same goes for this 3x3 grid here. This makes sense because the sequence cat is also repeated. This is really good to know because it tells us that there are different ways in which we could align the horizontal cat sequence and the vertical cat sequences. The third pattern we can easily identify are inversions. Let's fill in our matrix a third time for two different sequences. In this example, the most striking pattern is this diagonal here. It shows us that there could be good alignment between these two subsequences. However, this diagonal doesn't go from the top left to the bottom right, as diagonals in previous examples, but from the bottom left to the top right. And that is because the sequence was inverted. If you look at the vertical sequence from top to bottom, you get C, A, T, C, C, A, and you can get the same sequence from left to right, C, A, T, C, C, A. Looking at inverted diagonal patterns in the matrix can help us identify inversions in the sequences, which could, for example, be caused by mutations. To summarize, there are three different patterns that we can easily find with this method. Firstly, insertions and deletions through gaps. Secondly, repeats through repeated blocks. And thirdly, inversions via diagonals. This method doesn't always give us the ideal alignment. However, it is good preparation for learning about algorithms that are more comprehensive, namely the Needleman-Wunsch algorithm and BLAST. I will talk about these two algorithms in my next videos. Thank you for watching.